Hello, hello. Can you all hear me?
Morning.
Good
Morning.
morning. I can hear you.
Yes.
Good morning.
Good. So today we're starting on the cell. Next time we meet, we will be starting in and all of that chemistry. A number of you sent me email messages last night. I was trying to answer most of them. Did I get everybody, I hope? And if not, stay around near the end of the hour and we'll try to get through whatever issues are arising. We always end up having, growing pains is what I call it at the beginning of the semester. So as we all get used to this, and God knows every semester it's a new story as to what they're doing, isn't it? Okay, so let's get into the meat of this. We're going to talk about cells. Most of this will feel like a review because most of you have had basic biology probably pretty recently, maybe just before this class in college, or at least you had it in high school. All right, so let's get into it. So we're talking about cells, cells being the basic unit of the body, the smallest, most basic unit of life. And we measure our cells at the level of the micrometer, okay? Micro, okay? 10 to the negative 6, right? So nearly all cells have a complete set of genetic instructions, and this would be each cell basically containing the entire genome. But in each cell, especially as they differentiate, they don't turn on the whole genome and start making every protein that could be possible in your body. They turn on the little part of the DNA that they need to transcribe for that cell to do its thing. Okay, to make its proteins and function as its unit in the whole of the body. Okay, so it only needs to use a little part of the DNA. The whole genome includes the recipe for everything. So we just use a little small part of that genome in each cell. Cells that are specialized or are differentiated kind of are like terminal cells that ultra specialize and use a very, very small part of the genome. So they're the most terminally differentiated cells that we can have in the body, something like a nerve cell. All of our cells come in a variety of different sizes and shapes. You'll have really, really long cells, such as a muscle cell or a nerve cell. Say like a muscle cell that's in my thigh. That cell at the top of my thigh, all right, is going to be extending all the way from my hip bone to my knee bone. That's how long that one cell is. Okay, and we'll get more of a feel for that as we get into all these specialized cells. Whereas, like a teeny tiny little cell in my eye that's a photoreceptor picking up the light and telling my brain what colors I see will be a little teeny tiny submicrometer level kind of cell. All right. Now let's keep this in slideshow. Okay, so here you see a bunch of the different cells. You can see that the nerve cells will have long extensions, they're axons. So they too, like the muscle cells, will extend for a long part of the body. And all these axons will extend from the main cell body. And the cell body is where we'll have the nucleus and a lot of the organelles that are associated with the cell. Our skin cells are going to be organized in, pasted together, sort of flat sheet-like epithelium, okay? And again, talking about the muscle cells, they're going to be filled with, you see all these stripes in here, All these contractile proteins will be in the muscle cells that enable them to contract and pull together and attach to things like the bones, the ligaments, all right? And you can see different shapes of cells here, something that's a little bit more, I guess you'd say, parallelogram-ish right there, as opposed to a nice rounded up cell with a nucleus that seems to be bulging in the center. Nuclei are not always in the center, all right? It just depends on the cell. We'll see so many different arrangements. Okay, here is what we call a composite cell. And a composite cell is an idealized cell showing many of the structures that we have inside of a cell. You probably won't find any cell that has all the things that you see in the cell that's depicted here, all right? So we can see we have a nice big nucleus in the center of the cell, and in the center of the nucleus is the nucleolus. And anybody want to take a stab at remembering what the nucleolus does?
To have the chromosomes?
Wrong, but that's always the number one guess. Okay, so we're going to stop thinking that the chromosomes are in the nucleolus. Okay, so thank you for answering that, because that's what everybody wants to answer. So now nobody's going to want to answer when I say, what does the nucleolus do?
Does it program or program what the cell to do, tells the cell what to do?
Not really. So the nucleolus really is the center of housing the RNA that becomes useful in making ribosomal RNA. That's sort of its job, all right? It's for ribosomal RNA. Okay, so you'll see a lot of nucleic acid housed there, but it is not the genome of the cell, all right? So all this stringy stuff, do you see all this stringy stuff coming off behind the nucleolus? And you see the label chromatin going to that stringy looking stuff. All right, so chromatin, do you remember what chromatin is?
I do not.
Okay, chromatin is kind of the lacy, I almost said lazy, the lacy, unwound DNA, okay? When it gets wound up on the histone proteins and it contracts up, it becomes the chromosomes. So this is kind of like the open DNA, kind of living around in the nucleus, okay? So that is your genetic material, chromatin, all right? The nuclear envelope is a phospholipid bilayer, just like the plasma lemma, right? Okay. And the nuclear envelope has these little holes in it. Do you remember what those are called? These are called nuclear pores. And so when all this chromatin kind of opens up and we have DNA polymerase and RNA polymerase, some of our lovely favorite enzymes come in and hook up on there, DNA polymerase will make more copies of DNA for DNA replication. RNA polymerase sits on the DNA when the DNA is all nice and open up to translate the DNA into RNA. RNA, your DNA always stays in the nucleus. Your RNA is the messenger RNA that can shuttle out, and it shuttles out through the nuclear pores and kind of winds around and travels in through this endoplasmic reticulum till it finds some nice ribosome on this endoplasmic reticulum that it wants to jump onto, where that RNA message will get translated into protein, okay? All right, so this is our DNA the recipe for all of our proteins, all of our structures of our body. RNA polymerase can sit on that, travel out through the nuclear pore, take that RNA little message, little mini recipe, right? And it's only going to translate some of the proteins the cell needs. And it takes that recipe over to the ribosome. Let's pick this ribosome right here, sitting on our rough endoplasmic reticulum. All this endoplasmic reticulum that's contiguous with the nuclear envelope. This stuff is going to be studded with ribosomes, making it rough, okay? The endoplasmic reticulum on which you don't see any ribosomes, okay, right out here is called smooth endoplasmic reticulum, okay? And that will be more for being involved in things like maybe forming some lipids or that. But our proteins are made on the ribosomes. The proteins that are made on the ribosomes on the endoplasmic reticulum are usually the proteins that will then get passed on to the Golgi apparatus over here, the Golgi. And at the Golgi, they're going to modify the proteins that are made and kind of pinch some of those off into little vesicles like this that pinch off of the Golgi. And then the Golgi vesicle will travel over to the edge and release as secretory vesicles the proteins that are made. So these are often the proteins that are destined to be extricated or extruded from the cell. The proteins that the cell itself likes to use usually are made on these free ribosomes. You see all these ribosomes out here that are just kind of out in the cytoplasm. Well, when the messenger RNA travels out to these, it makes the proteins and they just stay in the cell and are used by the cell. Okay, so that's kind of the two ways that the protein gets trafficked in the cell. And I know this is going fast a little bit. We'll kind of come back around to this a couple of times. Okay, so the more you hear it, the more you'll get it. I like to kind of go through it with the pictures for everybody who's a visual learner. And then of course, we'll have things that are as well just written out. Okay. All right, so we looked a little bit at the nucleus here. We got the DNA and the chromatin, and we got the nuclear pores. We have the rough ER. We have the smooth ER. We have the Golgi apparatus. What other critters do we see in here? Let's see, this is a mitochondria. Anyone knows what happens in the mitochondria?
It makes the energy for the cell.
It makes the energy for the cell. Yes, exactly. So the cell is going to take up sugars. Okay. It's going to bring some sugars into the cell from, nutrition that you eat. It's going to bring the sugars in, lock it inside the cell. It's immediately going to phosphorylate glucose into glucose 6P. All right. Which traps it inside of the cell. So the glucose can't run back out of the cell. And then it's going to start acting on that glucose and undergoing glycolysis, all right? And we get to undergo glycolysis and we'll get some starter molecules out of glycolysis that will then shuttle into the Krebs cycle and the electron transport chain. And Krebs cycle and electron transport chain are going to take place here in the mitochondria. And what they're going to do is start extracting all these high energy electrons off of the glucose and ultimately shuttle them along this electron transport chain to make more and more ATP, somewhere around 32 to 36. This is all theoretically, so probably less in practice, ATP per molecule of glucose. And I probably just lost a lot of you there. You're like, oh my God, chemistry, and we're not even talking about that yet. We're throwing it out here. I don't expect you to remember that from this lecture. I'm not just saying it to show off that I know it, but we're trying to, from the immediacy, kind of see how we can put all of this stuff together, all right? Because pretty often, no one bothers to do that for you, all right? So that's our mitochondria. Also in here, you're going to have a bunch of little lysosomes, okay? Lots of lysosomes. Do you know what a lysosome is? Okay, so a lysosome is a little sac of digestive enzymes, okay? So things that the cell needs to get rid of, it's going to shuttle into a lysosome that can, chew it down. Maybe a toxin, maybe it's, something nutritional that it just needs to get breaking up and get digested before it can use it. So it's a sac of digestive enzymes. Oftentimes we can, even in lysosomes, when your cell gets invaded by bacteria or something, it can kind of shuttle that bacteria into a lysosome and start letting the lysosome digest something like that. All right. Okay, let's look at a bunch of other structures that we see here. How about these things? What are these out here?
Cilia.
Cilia, okay. And what do cilia do?
Don't they move the cell?
Well, they don't really move the cell so much, especially in the human body. Our cells don't really travel around. They have these microtubules, but they beat, okay? And so you think, well, why does a cell need to, kind of beat things? Well, a lot of things that the cells might make and extrude, let's say you're in the respiratory system, these cilia out here, they're going to put a bunch of sticky mucus out with the cilia. And in your respiratory system, you breathe things in and you want to trap all the particulate matter. You want to trap those dust bunnies. You want to trap allergens, things like that. And then the cilia is going to work in unison to beat it all up in one direction so that you hack up that loogie, right? So that's literally what cilia will do in the respiratory system. Cilia, somewhere like the reproductive system, will do things like beat and beat to move the sperm toward the egg. Okay, so that's cilia.
Quick question. So could I make a generalization and say the cilia is used to move particles around whatever's coming around it? Almost like a, I don't know, I don't want to say gatekeeper, but it's to whatever debris is coming around it, it moves it around. Okay.
Yeah,
Thank
cilia is going to be kind of like the cell's brush or broom, okay? And it's going to brush the mucus. Like I said, in the respiratory tract, in the reproductive tract, it's going to be helping to brush the sperm up to meet the egg, okay? Now, over here, you have something that looks like short cilia, and these are called microvilli. Anybody have a clue what microvilli do? And there's no shame in not knowing, okay? So this is why we're learning, right? So microvilli, they're very different from cilia. So cilia, you can see, have all these microtubules in them. That's what makes the cilia motile, okay? Motile in the sense of the cilia beats. The microvilli, however, their job is to be extra projections of the cell membrane to increase the cell's surface area so that food will get trapped down here in between all these little grooves in that. And there's more surface area contact for food particles to make with these microvilli. And it's at the microvilli that food absorption takes place. So the small intestine, which is the primary site where food absorption takes place, tons of microvilli. And in fact, you'll find microvilli on top of villi. So you'll have little stacks and stacks of these little projections on bigger projections. And so we'll get to that when we get to the digestive system. But again, it's idealized. Okay, and then we got this big guy out here at the end. Flagellum, okay? What's the flagella do?
Moves the cell.
It moves the cell. But I just told you, our cells are pretty non-motile in our body, right? So how many human cells do you think have flagella?
Just the sperm.
Just the sperm, exactly. And the sperm, basically, when we get to the sperm is going to get rid of all the organelles, okay? And it's just going to house its DNA. It'll have an acrosomal cap on it with lots of enzymes so that it can penetrate through all the cervical mucus and get to the egg. It's a lean, mean fighting machine, or I should say it's a lean, mean impregnating machine, right? So it gets rid of everything else in there. Solely, it's like a nuclear guided missile getting to the egg. It's got its digestive enzymes in the acrosomal cap, and it makes its way, and voila, pregnancy happens, right?
I have a question. So in other organisms besides humans, would there be other varieties of cells that have flagellum?
Yeah, well, especially when you get to micro, you'll see all kinds of bacteria, all kinds of protozoa have flagella because they swim around in their environment. You know, the bacteria swim around in the environment of your, fluids in your lungs and, try to swim over that mucus layer. And often they get trapped and your cilia help beat them back out and you cough up some of that, some of those bacteria. But bacteria are swimmers. Protozoa are swimmers. There's a bunch of other things out there, of unicellular animals that have flagella. But flagella only in the humans is only in sperm. Okay. All right, here's another idealized cell. I think I put this in before I loaded up the slides for you guys. And so I'll reload it at the end of lecture because I thought maybe you'd want a picture where you could just sit down and try to write what everything does by it, try to identify it, draw a picture from mitochondrion over to mitochondrion over here, kind of an arrow. And then you can write typical words you learn in elementary school, powerhouse of the cell, but you might also want to remember that the Krebs cycle and electron transport takes place here, okay? Nuclear pore right here, you want to remember, that's where the mRNA travels out to get in here into that endoplasmic reticulum, okay? So we're going to try to put it together in a much more dynamic way than we often learn. All right, so let's start talking about some of these structures now. So our plasma membrane. Our plasma membrane is thin, it's flexible. I wish I didn't have something that keeps beating on my, dinging here on my computer. I'm going to have to figure out how to shut that dinging thing off. So I apologize for that. All right. So the plasma membrane is thin and it's flexible, and its main job is separate the cell from its surrounding environment, whatever that environment is, and also keep all of its innards nice and contained so they aren't leaking out of the cell. Okay. The cell membrane is going to be made of lipids. It's going to be made of some proteins, and it should have a few carbohydrates studded throughout it. So the basic framework, you've always learned this probably, is a mantra, phospholipid bilayer, right? Phospholipid bilayer, phospholipid bilayer, phospholipid bilayer. Say it over and over again, never forget it. The cell membrane is a phospholipid bilayer. And we'll go in depth, especially as we get into the chemistry section, on exactly what all that means, okay? But we'll start a little bit here. So the phospholipid bilayer is made by phosphate groups that are bound to 2 fatty acids, okay? Phosphate bound to 2 fatty acids. And then you'll see one after another of these, and they all take on basically the same orientation in space to make a bilayer of these. I got a picture coming up. A picture will tell you more. Along with all this phospholipid bilayer, we're going to have, oops, told you that I got that trigger happy mouse. We're going to have a bunch of proteins that will be embedded in there. Some of these proteins will be things like forming ion channels or forming receptors to communicate with things outside the cell. So they'll be very important because, as we know, our cell is selectively permeable. It's only going to let in specific substances as they are needed, right? Okay. And
Like a bouncer?
Like a bouncer, exactly. And even though that bouncer might let, pretty girl in sparkly dress on Friday night, he might not let that same girl in on Saturday night. And the cell's the same way, even though it might let in, glucose today. Maybe if the cell's got enough glucose now, it's going to shut down and say, sorry, glucose, go stand in line somewhere else, right? Okay, so very good. Bouncer at the bar. Now, the plasma membrane is going to enclose this basic gel-like substance, which is called the cytoplasm, okay? So the cytoplasm is made of a liquidy gel called the cytosol, okay? That's the main liquidy part. And embedded in all that liquid of the cytosol will be all the organelles, the Golgi apparatus, the lysosomes, the peroxisomes, all that stuff, ribosomes moving around. And to communicate with the cell, there will be a fancy term called signal conduction. And this is what will allow the cell to receive messages from outside the cell or send messages somewhere outside the cell, or even communicate amongst the parts in the cell. And we'll get into signal transduction, but just kind of want to introduce it here. Okay, we'll get into it a little bit more, I guess, right now. Forgot that this slide was here. So signal transduction is basically the process that's going to allow your cells to receive messages from outside the cell and respond to messages from outside the cell. So cells will have, as I said, proteins embedded in that phospholayer bilayer membrane. Those proteins are often receptors, and they'll be sticking out, and it's almost like they're sticking out and they're going to catch balls that are coming to them. And those balls can be packets of information that the cell needs to receive. And so that it'll receive it, and then on its long arm that crosses into the interior of the cell, because this is just jutting on the outside of the cell, it can translate or take that bits of the message to the interior environment of the cell. Okay. All right. So we'll get into this a lot more later on. So your cell is going to have lots of different protein receptors embedded in the exterior. So your cell can receive lots of different specific messages from, say, other cells in the body. All right. Some receptor might be, out there for insulin. Okay. And insulin comes and sits on its receptor And insulin will actually be almost like a lock and key that can open up the cell so that allows your cell to take up glucose. Okay, that's kind of how insulin works. It's like the key that unlocks your cell to say, okay, you can take up some glucose. And we'll see how when that key isn't working so well, and you have, say, type 2 diabetes, we might have to have some drugs like metformin, glucophage, that help you push that key open to let the glucose get into the cell. All right. Don't worry, you don't need to know that quite yet. All right, so there's the cell. All right, so the plasma membrane basically isolates. Physical isolation is one of its main functions. The barrier between the outside of the cell and the inside of the cell. The cytosol is going to have a different composition or makeup of, all of its fluid level, how much sodium it has in it, how much potassium it has in. It's going to be quite different than what's on the outside of the cell. And so we can't have, all this exchange going on. We have to keep our interior of our environment of our cells stable. All right. The plasma membrane is going to regulate any exchange of these ions, such as potassium or sodium, with the external environment, because all your cells are bathed in all this extracellular fluid. All right. It's also going to allow the cell to extrude or get rid of the waste products that it doesn't want to keep inside the cell. That's part of that barrier. The cell also, through its membrane, needs to be sensitive to what's going on outside of the cell. It needs to have some way of kind of detecting, hey, is my environment changing? And do I need to respond somehow here in the cell to changes I can detect that are going on around me? And that's going to be, again, through binding a lot of receptors and translating a lot of signals into the cell as to what's happening outside of the cell. And we'll see that the cell membrane itself is providing structural support to the cell. So you'll see one cell will kind of anchor to another cell to start forming tissues. You'll also have a lot of microtubules and actin and things that will be right underneath the cell membrane, giving the cell its particular shape and its particular structure. Okay, so let's talk a little bit more about this phospholipid bilayer because it's so important. So the phospholipid bilayer is going to be made out of phospholipids. Okay, so it's going to have a lipid portion. And what do we know about lipids? If you throw a lipid, if you throw butter in a glass of water, what's going to happen?
It stays together. It doesn't.
Right, exactly. It stays together. It doesn't mix, right? It doesn't mix. Oil, you want to make salad dressing, you stick oil in water or oil in your vinegar for your salad dressing, it's probably going to float on top, right? Because it's both lighter weight and doesn't mix. All right. So we're going to have a lipid component and we call it hydrophobic. That means it's not mixing with the water. This is water insoluble. These tails, these fatty acid lipid tails, exclude water. And you can see that's going to be a very important part of the barrier of the cell membrane that keeps the water of the external environment out of the interior of the cell and also prevents the water on the interior of the cell from leaving. Okay. However, we can't just have this big fatty membrane that repels the water from the exterior and the interior, because we do need the exterior to communicate with the interior, and we can't just have some airspace all around that, right? So we have hydrophilic heads that extend from the surfaces of these fatty acid tails. All right. They're hydrophilic, so they interact with the water. Together, having a hydrophilic head and a hydrophobic tail, the whole entire, and here's what it looks like. I didn't mean to just do that, is something that we call, hang on, let me get down in here so I can do this. Draw, insert. When you have something that has both ends like this, we like to call it amphipathic. Okay, amphipathic means that it has both hydrophilic. Okay, let's change the color here or something. Computer's being really slow.
So the heads can take in the water, but the tails.
The head will interact with the water. The
Okay.
tail excludes it, avoids it. Okay. So amphipathic is both hydrophilic and hydrophobic. It's a molecule that has both parts in it. Okay.
Okay.
All right. All right, so this helps make the bilayer being permeable to some lipid solid substances, but impermeable to water soluble substances. And we'll see that when we look at the picture. You will also have cholesterol in the membrane. Cholesterol, big clumps of fat, kind of gets an evil name, right? That we should be avoiding cholesterol, right? But cholesterol is necessary for life. Okay, so no cholesterol, you have no life. All right, so don't think of cholesterol as entirely evil. Cholesterol is a part of every cell in your body. All right.
So for us middle-aged people, talking about myself, then that's when we talk about like good cholesterol versus HDL versus LDL.
Sort of. Cholesterol actually gets a whole lot more complicated than just thinking of HDL as good cholesterol and LDL as bad cholesterol. And I can direct you to some sources. But, you know, that's kind of like the simple thing. You know, we like to be simple-minded in most of medicine. So that's kind of the simple thing, and it holds for most stuff, but we'll get into it later on. Okay.
Okay.
All right. All right. So some lipids and proteins can move around through these phospholipids that are making up our bilayer. Our bilayer isn't just static, like you don't have a lump of cholesterol just sitting in one spot and all the bilayer and it doesn't move around. All this stuff is very, very fluid. Okay, so let's start looking at our bilayer. And move on to the next thing here. All right, so here is our hydrophilic head. This is what likes water. These are our fatty acid tails, all right? And you can see that this up here in our hydrophilic head has charges on it. And as you know, water is polar, right? Water is polar. And this is where we need to really go through and have our what do you call it? Our chemistry. Because if we remember, water is oxygen, right? And 2 hydrogens attached to it. And we'll go in through this in our next lectures. Our hydrogens have little partial positive charges, all hydrogens on water. And our oxygen has lone pairs of electrons. Just one circle there, okay, and another one. And our lone pairs of electrons, all electrons have negative charges. So this is kind of a delta negative or partially negative charge, okay? So our negative charges on these oxygens will be attracted to the partial positive charges on hydrogens, okay? Like charges repel and opposite charges attract. And so this is how water interacts with these phosphate heads, all right? Now, all these fatty acids down here, these are uncharged. They have no desire to interact with the charges on water, okay? So that's why they repel water. So then what you see is all these phospholipids line up, one fatty acid head after another fatty acid head, all these phosphate groups, and then all the long fatty acid tails orient in the same direction. And on the other side, the same thing happens. All these phosphates with all their charges line up. All the tails kind of orient to face all the other tails. These are water excluding. So all this in here, this all hates water. Okay, hates water. Hard for me to write with this mouse, I'm sorry. Okay, so this is all going to exclude water, doesn't want to interact with it. This stuff out here, these heads, loves water, right? Hydrophilic, loves water. Make that look more like a 2, okay? So when they line up here and form our cell membrane, you have all the heads out here loving water, interacting with the extracellular water, right? Water extracellular. Whereas all of this part in the middle is going to exclude things from traveling in through the membrane, okay? Hates water. Then on the other side, facing the interior of the cell, sorry, that's supposed to say interior. Interior of the cell. This is also watery, right? It's cytosol. Cytosol is all watery. So you have your hydrophilic charged phosphate heads facing all the water on the inside of the cell. So you can see phosphate groups over here interact with extracellular water. Phosphate groups here interact with intracellular water, and all those tails in between are what's excluding the water from moving from the interior to the exterior, or from the exterior to the interior, okay? And then you'll find at various points, we'll embed, you know, some proteins, some little receptors, right? That can interact. This will be a theme that goes all along. So when we have hormones that come and sit on a receptor, and they can send their little hormone message down here. Maybe there's a tail of something on here, and it can interact with some molecules in here, okay, so that a hormone can come in. We could have receptors that we have coming through that are for, you know, some other cell communication, like a neurotransmitter. Okay. We could have ion channels that we put through here. So we put a little opening, okay, And it can have a gate that can close off maybe and open up when we want it to, so that ions like maybe sodium ion can travel and get into the cell. All right, so we'll start building all these things in our membranes. All right, so here's a picture with a lot of stuff already put in it. So we have here some glycolipids sticking out. You got a big protein here with some, looks like chains of carbohydrates. So this would be like a proteoglycan. Okay, something like that, or you could call it a glycoprotein. Here's some carbohydrates, some more carbohydrates attached to a protein, carbohydrates attached to a protein. You have some transmembrane proteins that span the whole membrane. That looks to me like something that's going to be a nice receptor, right? It's going to maybe catch something right up here. All right, then you have some proteins that aren't spanning the whole membrane. All right, so we'll work through a lot of these. Here's a protein that has a channel in it, so you could see how things could get in through that channel. And it's probably exclusive to most things, but only lets certain things through, like small ions. All right. All right, so clinical application that was in your textbook goes through how you can have faulty ion channels. So when we go back, let's go back here like this. Here's an ion channel. Let's say in this case, it's an ion channel that just lets sodium ions through maybe. All right. So you can have channelopathies. So a channelopathy would be suddenly you have a channel that doesn't work. All right. Inherited erythromelalgia is a channelopathy, and it's a pain condition. And it's a channelopathy, and it's a channel for sodium ions. And so the affected individuals who have this channelopathy experience searing, burning pain, usually in their extremities, distal extremities. What's a distal extremity? You learned the word distal the other day.
Feet and hands.
Yeah, feet and hands in response to mild warmth. Okay, so it's pretty awful. Cystic fibrosis is probably the most common channelopathy there is. It's the most common genetic disorder amongst Caucasians in North America. So the CFTR gene creates a chloride channel, and this chloride channel is put in the apex of epithelial tissue throughout the body, and the channel controls the volume of liquid on the epithelial surfaces by secreting chloride and inhibiting the absorption of sodium. So this terrible channelopathy ends up causing really like thick mucus to build up in the lungs. Okay, so people with cystic fibrosis, and it can be a range of how bad the disease is by how much of the gene product is affected, get really, really thick mucus. You know, you see all these therapies and that for helping pound on the back of the kids and that to break up their mucus and get them to cough it up. But really the treatment is ultimately getting a lung transplant. But it also affects other organs other than the lungs. And the lungs is, of course, the primary thing that we always think of. It can affect the sodium and chloride exchange in the pancreas and in the gallbladder and in the reproductive organs. And so oftentimes there are pathologies going on in many of these other organ systems as well.
So, and you are saying these disease processes all kind of originate at the cell membrane level? There's some kind of dysfunction with the, okay.
Yep. So, we look at all these ion channels and I think people always say, oh yeah, ion channel. Okay. Next, but there really are serious diseases that can happen with something as simple and basic down to the cellular level of the ion channel being defective. So when we get into studying the heart, this is just kind of a little preview. You'll see that in our heart, we have ion channels that control the rhythm of the beats for our heart. At various times, we first let in sodium ion channels, and that allows the depolarization to start the beating of the heart, okay? And then we'll have potassium exit our cell, and that starts to repolarize the cell to make it stop beating, okay? And then we also have calcium come in to allow a sustained beat, because you don't want your heart to just beat, close. You want it to beat and allow it to kind of have a squeeze so that you have blood filling up the chambers of your heart for a while. And then you need to stop you need to repolarize, okay? If that makes any sense to you yet, it might not. So anyway, it's allowing the beats of your heart that allows ultimately the squeezing the blood out of your heart and the opening back up for the filling of your heart. That's what your heart does. Fill with blood, squeeze the blood out. Open back up, fill with blood, squeeze the blood out to pump the heart around your whole body. When the beats, the rhythm gets off, then you have problems with either squeezing the blood or filling, okay? And this is what causes, major cardiac dysfunction. So these can actually happen also at the channel level. Now, both of these slides are just kind of to get you start thinking. Don't sit around and try to memorize and understand these diseases yet, okay? This is just put in here for you to kind of start thinking like, oh, okay, and encourage you to pay attention to these channels because they are actually important. All right, now when we're also talking about cells, we have to talk about cellular adhesion molecules, CAMs, okay? CAMs will guide cells that are moving. And one of the primary moving cells that we have are our blood cells, white blood cells and red blood cells. And so you see over here in a blood vessel, right here, we have a bunch of white blood cells, white blood cells, white blood cells. And they're trying to move along this blood vessel, all right? But sometimes we need to pull our white blood cells out of the blood vessel where they're traveling around and get them into an area of tissue where maybe there's infection, because our white blood cells are cells that combat infection. They're our immune cells. And so we have these various cellular adhesion molecules that will kind of attach to them, kind of stick to them, and kind of pull them off, where they need to go. So one of these here you see is a selectin. And a selectin is a type of cellular adhesion molecule along with other cellular adhesion molecules. And in this case, they're getting the white blood cells to marginate or stick to the margins of the blood vessel. And then they even help open up spaces between all the blood vessel cells so they can undergo what we call diapedesis, which is literally this white blood cells walking through the endothelial layer so they can get to the site of infection. And you see here, there's a splinter going on. So you know that somebody's gotten a splinter and it's gotten through the skin and into the blood vessel. And there's going to be a lot of inflammation and infection going around that splinter. Okay. So that's how cellular adhesion molecules work. Now our cytoplasm, when you think about it, you always just sort of, oh, it's the jelly-like substance that's inside the cell that kind of all the organs are deposited in there. But it too has its particular composition of electrolytes and, and it can change from cell to cell. So your cytoplasm is the actual gel with all the membranes and organelles in it, okay? The cytosol is the fluid jelly-like portion of the cell, okay? And then you have all the organelles, the mitochondria, the Golgi apparatus, et cetera. They have their specific functions in the cell that are embedded in the cytoplasm. Also in the cytoplasm are all these fibrous substances, which are the cytoskeleton, all these like microtubules and microfilaments that help give it some structure. And you'll see a lot of that is really right underneath the plasma membrane. Okay, so your cytoplasm is part of what we call the intracellular compartment. And about 2/3 of your cytoplasm is going to be water, because 2/3 of our human body is water. The remaining 1/3 is found in what's called the extracellular, excuse me. I'm gonna have to cough. Sorry. The remaining water is going to be found in the extracellular compartment, which is going to be interstitial in between all the cells of the tissue, or intravascular, actually in the blood vessels, or transcellular, just moving across the cells. Hang on one second. Sorry, had to really cough. I didn't want you to have to listen to that. All right. All right, so getting back to some of our organelles, ribosomes. So ribosomes are made up of protein and RNA. The RNA that it's made of is housed in the nucleolus. All right, we have ribosomes that are free in the cytoplasm, or we have ribosomes that are associated with the rough endoplasmic reticulum, okay? And they, sorry, God, I got to do something about this mouse. I think I might have to drag out my old mouse and hook it on here. The ribosomes will also provide some structural support, and the ribosomes can have enzymatic activity, in which case they're called ribozymes, okay? And they help link together our amino acids in protein synthesis, making the long chains will go through protein synthesis, all right? The endoplasmic reticulum you see over here, all right? And it is continuous from the nuclear membrane. And it's a series of membrane-bound sacs, canals, or vesicles. And it's got this inner sort of tubular system that will transport things like the mRNA, and will transport the forming ribosomes as they come out from that nucleolus. So the rough ER is a conduit, right? The smooth ER over here doesn't have the ribosomes associated with it. It's not studded with ribosomes. So rough ER, making proteins on all those ribosomes. The smooth ER is more for lipid synthesis. You're going to have various vesicles throughout the cytoplasm. A lot of membranous sacs. You can see some of them will do things like pinch off the Golgi apparatus, okay, to export proteins that were made to the exterior of the cell. Sometimes you'll see we pinch things, pieces of the cell membrane off, and take things into the cell, and that little piece of cell membrane will end up being a kind of a sac that's moving around in the cell to deliver substances that the cell took in from the outside. We have our Golgi apparatus. This usually is associated with the rough ER, because the rough ER kind of moves the proteins it was making over to the Golgi apparatus, where the Golgi is going to package up those proteins, and it might make some alterations to them. And like I said, likes to deliver it to the exterior of the cell. All right, so an example of doing this would be milk secretion, for feeding the baby, all right? And so you would make the milk proteins, all right, on the ribosomes in the rough endoplasmic reticulum, package them off and send them off through the Golgi, package the milk proteins in pieces of the Golgi, the sugars right here, okay? And some more milk proteins. The lipids come from the smooth ER. And so you kind of combined all of this at the end of the nipple, where you would end up extruding all those substances, okay? All right, the mitochondria, here you see a nice big blown up colorized transmission electron microscopy picture of it. You have mitochondria kind of throughout the cell. The mitochondria are housed with its double membrane, but inside it has this leaflet of membranes called the cristae, the inner membrane, okay? Now, the mitochondria is where the Krebs cycle and electron transport will take place. Glycolysis takes place out in the cytoplasm. And all this is going to happen along these leaflets, where eventually you'll see a whole bunch of enzymes are embedded in those leaflets that will extract the energy from the electrons that are delivered there. And this is what will ultimately lead to the formation of ATP. So if I look at a cell and I see a cell that has lots and lots of mitochondria in it, what does it tell me about that cell?
It makes a lot of energy.
Makes a lot of energy, probably because it might also use a lot of energy, right? Okay.
Is that also like, if you saw a lot of mitochondrions that doesn't make sense for that cell, is that producing, or like, let's say, is that how you can tell, like, this might be too far of a stretch, but like ATP, I know is like how your muscles like show stress, right? And so if there's a lot of mitochondria there, could it suggest possibly that like there's an injury or highly used?
No, highly used. So you'll see like if we took biopsies of someone who is a long distance runner, they are going to have a lot of mitochondria in their tissue, in their muscle tissue, as compared to me.
All right.
Or even versus a short distance runner, because a short distance runner actually burns up fuel through glycolysis. And we'll get into this later on when we talk about slow twitch and fast twitch muscle, red muscle and white muscle. And red muscle is the slow twitch for the long sustained activity and has therefore lots of mitochondria in it. So yeah, so you're thinking down the right path.
Okay, thanks.
Yep. All right. Some more cytoplasmic organelles, lysosomes. So lysosomes, like I said, are membranous sacs, and they contain tons of enzymes that can digest proteins or carbohydrates. They can digest, pretty much everything the cell encounters. They'll digest worn out parts. They can digest bacteria that infect the cell. They're the garbage disposals of the cell. And so it's very important that they have a membrane to contain all these digestive enzymes, because you would not want these enzymes leaking out into the interior of the cell. You would be auto-digesting your cell, right? So it needs to be compartmentalized. Same thing here with peroxisomes. These are other sacs, and they contain peroxide. And we'll go through hydrogen peroxide and what it does in the cell later on, but is very, very important for killing of bacteria, things like that. Then we have the cytoskeleton. The cytoskeleton is going to have all those things like microfilaments and, that you see down in here. And like I said, you see a lot of the structure of all these filaments is right underneath the plasma membrane. So they're these thready-like structures. And a lot of these will also become important in specialized cells like muscle contraction. We'll see that later on. Or the microtubules that will be in things like cilia or flagella, okay? The centrosome or the central body is made up of these cylindrical microtubules. And these will be really important when we get into doing cell division, because they line up and help distribute the chromosomes, okay, and help form like that whole complex with the centrioles. Cilia, as I said, have microtubules extensions in them. They form a little fringe on cells, and we'll find them in places where it's really important to have like this power stroke beating to either like beat up the mucus so that you can expel it, or to move the sperm and propel it toward the egg. Also move the egg down the fallopian tube toward the uterus. There's cilia there. Flagella is only in the sperm. You see here the sperm cell, and the sperm cell becomes, when we look at the interior of it, very highly structured and specialized. I said it gets rid of pretty much all the organelles and just becomes a little motile unit to deliver DNA. Okay, so here's some other things about diseases at the organelle level. You can have problems with your mitochondria that can lead to this mitochondrial encephalopathy that goes along with lactic acidosis and stroke-like episodes. This is called Malus syndrome. And there's a mutant gene in the DNA of the mitochondria. So you can't then extract all the energy from the nutrients you take in, this Malus syndrome. Now, interesting enough, your DNA, normally your chromosomes, you get, half your DNA there from dad, half your DNA from mom. But as we'll learn down the way, all of the DNA in your mitochondria, you inherit from your mother only, okay? Mitochondrial DNA is inherited from the mom. So remember when I said the sperm extrudes all their organelles? One of the things they extrude is their mitochondria. So it attacks and, penetrates the egg. The egg is sitting there with its mitochondria. And then as the sperm and egg unite and then later develop, all the mitochondria start out from the egg's mitochondria. So it's a completely maternal line. And this, when we're doing all these modern DNA testing, and they can, track the heritability somewhat maternally, it's all with mitochondrial DNA. Okay, so I thought that might be interesting for you. We know all the Y chromosome stuff inherited is a patrilineal line. So we can also find a bit of a matrilineal line through the mitochondria. Adrenoleukodystrophy is caused by a lack of a protein in the membrane of peroxisomes. And because of this, fatty acids can build up and destroy the myelin sheaths of nerve cells. And then the nerves are unable to transmit rapid impulses, rapid action potentials. And so you'll understand that better when we get to the nerves and we see how the action potentials are generated and how the myelin sheath that's formed is what insulates the nerves, so that, you know, kind of like around your big power cords, you always have insulation around the wire. The myelin does that. So if you have a mutant form of the fatty acids in the myelin, you're not insulating well, so you're not conducting very well. Krabbe disease. This is a disorder that results in the loss of myelin, a demyelination disease. So again, you're losing that insulation around your nervous system. You're losing rapid impulse or transmission of your action potentials, okay? All right, let's move on. The nuclear envelope. All right, so now looking at the nucleus, the nucleolus, all the chromatin in there, we have our nuclear pores. So it too is a double layered membrane, has the pores so things can travel in and out. And we separate thus our nucleoplasm from the cytoplasm. Somebody's got their microphone on. Can you turn it off? Because we're getting a lot of feedback. In the center is the nucleolus, where everybody wants to say the DNA is, but you're never going to do that now, right? Okay. It is a dense body of RNA and protein, and it's where the ribosomes get made. All that loose, all this loose, lacy extensions from surrounding that out in the nucleoplasm is the chromatin. And this chromatin is where the cell's chromosomes are. And, you know, they condense up and we see them as actual chromosomes when the cell is getting ready to divide. All right, so we're going to talk about movement of materials in and out of the cell. We'll talk about some passive processes. These do not require energy, do not require ATP, and that would be diffusion, osmosis, facilitated diffusion, and filtration. And then we'll talk about some of the active processes that will require energy, that use ATP to get things in and out of the cell. Active transport, endocytosis, exocytosis, and transcytosis. So diffusion. Diffusion is when we're going to move atoms, molecules, ions, whatever particle we're talking about, from an area of high concentration to an area of low concentration. Remember yesterday when I said, always remember, it's like that spraying that perfume at the beginning of the room and thinking of all that spread of the perfume as the spread of the molecules in a closed contained space of a room, a big giant box. If you spray at one corner from high concentration where the sprayer is, everything will try to equalize or distribute to be every molecule in equal amount of a part. They socially distance, right? Okay, this is chaos. It's through chaos, all right? Now, all of these atoms and molecules are in constant motion, and they don't want to keep running and bumping into each other. So that's why they distribute out as far and wide as they can from each other. So it's everything automatically will equilibrate. You see that here as the same thing with dissolving a sugar cube in water. Here you have the big lump of the cube. Sorry. but because the cube is in water and it's soluble, all the particles, all the molecules of the sugar, each sugar molecule, can dissolve or break apart and move into the water. And they're going to move as far apart until they equilibrate to exactly being all the same distance apart. And this just happens automatically. There's no energy required. This is simple diffusion. Okay, so now if we apply diffusion, but we put a membrane in here, and we say the membrane is permeable to both the solute, okay, and the solvent, which is water. Solute, let's call these molecules sugar molecules, okay? So the solvent will be the blue water molecules, and the solute will be the sugar molecules of red. All right, so we put a permeable membrane in here, and we see that on this side A, we have more solute of sugar, and on the side B, we have no sugar, but we have lots of water molecules. Permeable to both, so each should cross the membrane. And indeed, this is what happens. As you see here from side B, the blue water molecules are moving over to A, and from side A, the sugar molecules are moving over to side B until finally they equilibrate and we have the same amount of molecules on each side of the membrane. Okay, in the cell, this happens as facilitated diffusion, okay? So when with the cell, particles want to diffuse, say, from the external environment into the interior of the cell. And often they use membrane channels which control the diffusion of the molecules going through. Again, it's diffusion, they're not using energy. It's just that it's facilitated. It wants to move down its concentration gradient where we have, let's call this gray molecule, let's call it sodium. Okay, high sodium on the exterior of the cell, low sodium on the interior of the cell. So sodium is driven to move into the cell. And it's just kind of slowed down yet facilitated by having to make it through this membrane channel. All right. Osmosis is a special case of diffusion. Osmosis just refers to the diffusion of water in water alone. Okay, so osmosis is just diffusion of water. So we're talking about the movement of water across a selectively permeable membrane. So water molecules being blue over here on side A and on side B. And then we have some non-permeable, some substance that can't get through the membrane. And in this case, the example is protein. So we have lots of protein on side A. We have a minimal amount of protein on side B. So what's going to happen then is water can move and try to equilibrate this. So from side B, water is going to move and keep moving and moving up this side of the column here until everything is spaced out to look like there's an equal amount of water and protein to what's on side B, okay? No ATP is used, no energy is expended, only water moves. This we can use to think about osmotic pressure, okay? Because water can move in and out of our cells. So the osmotic pressure is the ability of the diffusion of water to be able to generate enough pressure to lift a volume of water as when the concentration of that impermeant substance is increasing. So we'll take an example here with blood cells. Here's a normal red blood cell. And we place it in what's called an isotonic solution. Isotonic meaning it has the same concentration of water and all of its solutes on the exterior of the cell as on the interior of the cell. So what happens is water moves into the cell, but water moves out of the cell at the same rate. So there is no net diffusion of water. All right. Let's see. Let's draw it on here. Okay. Water moving into the cell. But water's also moving out of the cell. And they do it at the same rate because it's the same concentration on both sides of the membrane. Let's say it's the concentration of sodium chloride in solution. Sodium chloride in solution. Isotonic saline, like what you would get as a saline IV. Okay. Here on the second one, we have the cells in what's called a hypertonic solution. This means we have a really high concentration of sodium chloride outside of the cell, okay? So it's high concentration. Inside of the cell, we have a low concentration of sodium chloride. Okay, so much less water, right? I mean, much more water in here, much less salt. Here, you have much less water, lots of salt. So it's going to try to equalize by having water leave the cell. Water is going to leave the cell.
So hypertonic draws the fluid out of the cell, draws the
Right.
water out of the cell.
Exactly, hypertonic. All right. We'll go over this a little bit more too. Hyper. Okay. And here in this last case, we see the reverse. We see a cell that has a high concentration of sodium chloride inside the cell. Okay, so it's low water. Low water. And so this is exterior to the cell is hypotonic. Hypotonic. Okay, and hypotonic is going to mean it has low sodium chloride relative to the cell, but it's going to have lots of water, right? Lots of water. So since water is the only thing that travels freely through the cell membrane, water is going to move from its area of high concentration into its area of low concentration. So the cell is going to swell up with water to the point it might even burst. Here we call this crenation. Okay, crenation. Crenation. Oh, I wish I need an eye pen, I think. Okay, crenation. And that's the cell shrivels up as it loses water. And you can kind of think about this in the sense of like, if you were making Kool-Aid, right? Okay, so you make Kool-Aid and you really, really like that cherry Kool-Aid flavor. So you get your cup of water and you mix it in and you mix it really strong to get a really, really strong concentration. Okay. Now, if you had some sort of beaker or something where you could have it with a permeable membrane next to water, that Kool-Aid would keep dissipating all the Kool-Aid into the next area of water until everything equalized, right? Okay. And that's what's trying to happen here, is it's always trying to equalize to make the concentration of sodium in water the same on each side of the cell. Here, you're fortunate when you have, I should write it, just a text box here would be easier than reading. Oops, darn it. Wanted to get the text box to write, this is isotonic, right? Isotonic, same concentration on both sides, okay? Isotonic. Hypertonic, higher concentration of sodium relative to a low concentration of water. So water wants to leave the cell to try to make this outside environment the same concentration as what's in the cell. Here you have hypotonic, low concentration of sodium chloride, but lots and lots and lots of water. And so water wants to move into the cell to kind of equalize its concentration with the outside environment. And the cell swells up and can even burst.
So
Okay. Oh, go ahead.
I was just going to ask, from a clinical scenario, when are patients given hypertonic and hypotonic solutions?
At different points. So, so sometimes, let's say a patient comes in and has a profoundly low sodium level on their metabolic panel. You're not going to give them isotonic sodium chloride because that's not going to help raise up their sodium level. And it's really low sodium level is very dangerous. You're going to give them what we call hot salts. So you're going to give them a concentrated sodium chloride IV, but you're going to have to do it super carefully and really monitor it, because there's some dangers in giving hot salts too rapidly. You can cause what's called pontine myelinolysis and destroy some of the myelin sheath on the brain. So it's very, very cautiously. In some dehydrated people, especially like babies coming in, oftentimes we give hypotonic saline. So we'll give them hypotonic saline, but we also might hang like a bag of dextrose with it. And it gets to be a real fine art in deciding some of these different.
Sounds very complicated.
It does. It's a lot more complicated than what you think. But, you know, in general, most people will just come into the hospital and they'll get a bag of, isotonic saline solution. It's what you give in most instances. And surgeons, they all like lactated ringers, which is also an isotonic saline solution, but it's got some other minerals in it. And it's just because of their training is what they prefer. I don't think one has been shown to be better to the other for any good reason. It's just habit.
I had a quick question. So the cells are reacting to the substances around them.
Yes.
Meaning that it doesn't always do the right thing, in that, like, if you have a lot of sodium in your blood and the cell wants to imitate that, it'll say, bring in the sodium, shrink the water. Am I understanding that correctly?
Well, remember, your sodium has to be transported in by carriers, right? It has to go through an ion channel. So what we were talking about here is the actual diffusion of water.
Okay.
Sodium is not moving, but we're kind of, in essence, equalizing the concentration of sodium on each side of the membrane by moving water. Yeah.
But like in
Here.
concept, am I correct in that it's trying to mimic what's there?
Well,
Not
they want
necessarily.
to equalize.
Yeah.
Yeah,
Okay.
They want to
But not-
equalize.
But not necessarily get to like the correct amount.
Right.
Okay. That's what I wanted to make sure
Yeah.
I was understanding, because I always thought that they would be doing like not but
Like
correct.
hit a point and say, stop, I'm good now, right?
Yeah, like I don't know why, but I assume that the cells would be like, oh, well, we actually need more water, so we're gonna do this. No, it's just reacting to what's there.
It's just reacting because the membrane is permeable to water, so water can move in and out. And
Yeah.
if the concentration is really high inside the cell, the water of solutes, the water will move in.
Right.
If the concentration of solutes is low, water will flow out.
Okay.
Yep, because we're just talking about osmosis, flow of water.
Flow of water.
Yep,
Okay.
flow of water. Yep, not the flow of the solutes. Okay. All right, so filtration. So part of why we care about all this movement of water is because we have various filters in our body. Okay. And so filtration works with these osmotic forces. So it's going to be the process that's going to force molecules through membranes by exerting pressure. And we normally kind of do a little bit of a lab on filtration, but it's one of the things we're skipping. And I think it's 11:30 now, so filtration might be a good place to stop. And we'll do Thursday lab today, and then we can pick up with this filtration and also get into our chemistry on Monday. Okay. Sound good? All right. I shall see anyone who's coming back for Thursday lab in an hour. Okay. All right.
Yeah, I have a really quick question.
Yeah.
Are we filling out the entire PDF for this? Because my teacher last semester was just doing like certain questions.
We're going to
Cool.
fill in pretty much all the PDF. We'll do a lot of it together today in lab. So
Okay, perfect. Okay,
Yeah.
yeah, because I was going through it and working on it a little bit just to
Okay.
get an idea. But
All right.
okay, yeah, thank you. I'll see you in an hour.
And weren't there a couple of people who wanted to hang on and ask me some questions?
Yes.
Yeah.
Yeah. Yeah.
Hello. All right. Okay, start shooting them at me.
I just have a quick question because I guess the syllabus, some of us are just a little confused on how to, I guess, prepare for class before lecture and like take notes, because it says that we were supposed to learn or start the diffusion stuff next week, but we kind of started it this week.
Well, it's just that diffusion, we're going to do diffusion stuff next week. It's just that there's some of it that's in here with this cell chapter. So we'll really go through diffusion and osmosis and all that next week. So here, let's bring up the syllabus and the schedules and try to go over. I know it's confusing. When they told me how we were doing this is why I ended up making this. We'll look at the schedule first. And maybe I wonder, can I share two documents at once to put A and B up alongside each other? I wonder. I don't think I can. All right, so we'll look at this. Let me share the screen. And maybe kind of go through some of this. All right, so this is the A schedule, all right, for all of you who are doing the A schedule. A schedule means that your student number ends in an odd number, okay? And then I'll show B schedule means that your student number ends in an even number. And now there's been a couple of people who've asked me, can I swap? And I of course will let you swap, but I need to have relatively about half the class at each session. All right, so yes, we were going to start the chemical basis of life, but I wanted you guys to watch some of those videos first. And so I said last time that we were going to start a little bit on cells today. So we're going to do cells today, and we'll do chemical basis of life on Monday, because chemistry is kind of the hardest thing, and we still have some people filtering in and out of class. So I kind of moved it around, but I didn't move it around on the schedule. So I apologize if that was confusing. All right, so today in lab, so A schedule means for the whole class, we're going to be on an every other week schedule, because we need to be socially distanced in the lab. So we will have half of the class come per the A schedule and show up in person when there are yellow boxes on your schedule. All right, so anybody whose student number ends in an odd number will show up in person next week. On Tuesday, if your lab is on Tuesday, on Thursday, if your lab is on Thursday. All right, so basically I'll have a quarter of the whole lecture class on Tuesday or on Thursday. All right, if you're on the B schedule, let me bring that one up. Hang on.
So do you want us to have the pre-lab chapter 7 done before we show up to lab next week?
Partly, hang on, let's get to it. I'm trying to keep my brain straight here because there's so much to keep straight. Let me show the B schedule. All right, so B schedule. Okay, so now if you look at the B schedule, you'll see that this box that was yellow for the A schedule is orange. This means, did somebody just say something?
Yeah, the screen is kind of still froze on A.
It's still froze on A?
It's in half.
Okay.
of A.
Is that now on B?
Okay, now it is. It's good now.
Okay. So you'll see where you had the yellow box for the A schedule for labs 3 and 4, you have the orange box. Okay. So B schedule, even number. Tuesday and Thursday. Okay. 1/4 of the class will be there on Tuesday, 1/4 on Thursday. This means you're going to stay at home and you can work on answering all those pre-lab questions that someone was just asking me about when you're on your orange box day. Okay, so on your orange box day, you'll have 3 hours at home. You can meet with your fellow lab mates in person if you want. You guys can meet online together. I'm going to be in with whoever is the yellow box class. So I won't be meeting with you when you have these orange box days, except for the orange box today, that we're all meeting. All right. So yellow box, going back over here to the yellow box A. Hang on, let me share over here with yellow box A. Okay, going back. So everybody with yellow box, skeletal labs, you'll meet in person next week. We'll go over the bones, all right, all the bone labs. You will then have an orange box the following week. And so you see, you'll be working on 2 actual labs, labs 3 and 4, skeletal 1 and skeletal 2. So you'll have this week's opportunity to see all of the bones. Then you go home the following week and all those questions that are on the lab, you can write them up, all right? And then everything will be due on February 7th, okay, the following week. Now, if we go back to the B people and we look at that, you can work on answering all those questions that are on those lab papers during this week, and then the week that all the A people are at home answering all those questions, you'll be in the lab and you'll be playing around with all the bones and seeing all the bones and how they fit together and learning the bones. Okay. So that's how it's going to work. Does that make sense to everyone?
I think so.
Okay. All right.
I think it does.
So let's say your Tuesday lab, you're scheduled for Tuesday lab and you end in an even number. That means you will come to lab in person on Tuesday, not next week, but the week after. If you are signed to be in Tuesday lab, but your at number ends in an odd number, you come next week, next Tuesday. Okay.
Okay, I'm kind of confused on that because the schedule that you emailed me is a B group schedule, but when I actually think about the even and odd numbers, my ID number ends in an odd number, which would be A group.
Okay, well, I posted all of them on
Did you? Okay.
online. It's
Okay.
online on Blackboard under the syllabus. Hang on.
Okay, I was just looking at it from the one that I got emailed, so I just assumed that was the one that I was supposed to print. So.
No, I thought I emailed both of them, but
Okay.
to everybody, but maybe somehow yours didn't attach.
Okay. Thank you.
Yep. But if you look up in Blackboard. Okay, let's get back to sharing again. Okay, over here on the button that says syllabus and schedule, I have the whole syllabus and I even put it out in the box so that it's just easy to go back to and read the syllabus. And I would have done it for the schedule as well, except of course we have this crazy 2 schedule system. So
That's got to make it difficult for you to organize all this.
this is why when everybody kept saying, can you turn on Blackboard and get the syllabus? They told us at the last minute and we all had our schedules for 15 weeks. So now we had to change everything to 14 weeks and we had to change it to this A and B lab schedule. So yeah, it's crazy. So I think I found a better way than most people because I like to be hyper organized.
I have a quick question. So right when the pandemic started, I bought all the lab stuff for the 2019, 2020. So that is the lab manual I have. Is that
Does it say
still
by Esther Grossman?
good? Yes, it does.
Yeah, that's the lab manual that we're using. So you can use that if you want to, or
Okay.
if they can
Print
take
them
it,
off.
or if they can take it to the if you can take it back to the bookstore and get your money back, I'd recommend doing that.
Oh yeah, I can't, but thank you.
Okay. All right. So here is where on the on the Blackboard, you can see I have schedule A and B, so you can download A or B. Okay. For those of who are looking. All right. So labs, when we get into the labs, this is what we'll be doing today. And I was going to go over this in lab a little bit more. So lab should show up for you as lab 1 and 2, membrane transport and body orientation. So what you'll do, you can click on your labs over on this side, it'll lead to this. And it says lab 1 and 2 instructions for membrane transport. This is all the stuff that we're going to be doing this week. Lab 1 and 2 body orientation, Grossman lab manual. You can click into here and download this lab. Okay. And this is the lab that would be in your lab book that you purchased, but none of you need to purchase it. Okay. I have them all loaded up on here. And what I did with loading them up on here is I've changed a couple of things around in some of the later labs, but you'll see that I have fillable boxes so that you can just type in as you're doing it and turn it in through Blackboard anatomical position. standing, erect, palms facing out, like that. Go on to the next question. So you don't have to write it all out by hand. Now, if you want to use your lab manual and write it all out by hand, you can do that, scan it in and turn it into Blackboard that way. I'm fine with that. Or if you want to hand it in to me physically at lab, you can do that too when you're at lab. Okay. But you might not be done with it then. Now, the one thing I was going to tell everybody is, you see how right here, I'm typing this in and I'm still in Blackboard. Don't do that, even though I just did that, because it's not going to save and you can't load it back up to the website. So what you're going to need to do is take it, click on it, download it, somehow. I forgot where my thing is. I got a little arrow thing here. Click on the download and then it should show up in my downloads. Fill it in on your own computer. And then when you want to go and save it to turn it in, because we always have a little problem with this. I got to move all this stuff around. Is maybe where it says 2022, maybe just put your name when you save it, so that's the version in which you filled in all your stuff. And then you'll load that up where it's like write the submission and you have to submit it to me, and you can click on all that and submit it back to me through Blackboard. It's kind of hard for me to do it because I can't really submit it to myself and show you here. All right. So that's what you're going to turn into me. And then I'll release lab answers after everybody turns theirs in. Lab 2, this folder in here, is going to contain all of your links to go to the Connect work. So getting used to Connect. It'll take you right here. And so it can show you already 6 students have started their Connect work. 27 people have not. 4 people have already submitted it. So pretty good, right? For the 35 of you.
That's those modules.
That's all the modules in Connect. Yeah. So that's one way you can get into Connect, and I can click to return to Blackboard. The other way is you can also hit on McGraw-Hill Connect. Go to my Connect section. I hope you're all seeing this, right?
Yes.
And then you see up here, there's all these lecture practice things, these folders, these are these APRs. I'd recommend doing them. They're very good, and people who do them get really high grades in the class. But
they're
all
pretty,
this,
useful. Yeah.
Yeah.
I like doing them.
Okay. All of this is gravy though. Okay. And you're not graded on that. What you're graded on are the things in these lab folders. And we didn't do lab 1. So lab 2, you'll see this is the exact same things that, I clicked into earlier, lab A diffusion, where we have, some people have done it already. And I can click back and go back to Blackboard. I can click on here and go back to Connect. And if I look over here, this is where your ebook is. So you should be able to click in here and look at the ebook. You have a practice atlas that's available for you. So you can go in there and study with the atlas if you wanted to. Lots of good stuff. The Connect stuff really does, it's really good. It really has helped bring people's grades up. It brings people's grades down in the sense that if you don't do it, you're not turning in the labs and the homeworks every week, and those 8 points across 14 weeks really adds up and will lower your grade. But doing all the Connect work, as I'm always saying, the more you engage your brain with the activity of answering questions and that, Connect does a lot of that through all those exercises, and it's really useful. And I am a great believer in it now. I wasn't so sure when we first started using it how good it was going to be, but I have now used it across 4 semesters, and the people who really use it get the highest grades in the class. So I recommend it. And, you know, you can sit around and read your textbook and read your textbook. And I like reading a textbook, but it doesn't engage fully as many firing neurons and answering questions and doing problems is really what makes you retain for the long term. And this is what does it. Okay.
So I have a question.
Yep.
This is my second time taking this course, and I think I still have Connect for the next few months active. So can I still use my previous Connect account with your class, or I have to register for your class separately?
You probably can, and you'd have to tell me what section you were in, so I would have to go looking for your work. But the Connect stuff is supposed to be part of your tuition and free because they're piloting some project here. So you probably have a Connect account just for signing up for this class. Because they're piloting a project through McGraw-Hill, and we agreed to participate in the sense that they would give our students this free service of getting Connect. And so they want to integrate the ebook with all the questions, and they want to see how you guys do in the end. And so you probably have it in two ways. And I've had students who had 2 Connect accounts in the past, and both of them showed up in my class ultimately. So, you know, we can figure out how that happens, you know, whichever one you start working on, Benita. I can always go in there and search for your homework somehow.
Okay, all right.
Yep.
I'll look into it, and if I can get around it, I'll try to reach them, and then I'll email you too.
Okay, and I posted in a section for how to the technology people at Connect. I posted a section on that little you know, blue navigation panel. Plus I sent you all out an email, so you should be able to deal with your technical issues with Connect through them.
Okay. All right. Thank you.
Okay.
To add to what Benita asked, she should be able to, because I took this class before. I used the same book, and all I did was put in my email, and it gave me access to your class after I clicked the McGraw-Hill link in the Blackboard.
Okay.
Oh,
good.
Okay. All right.
Great. Thank you
I'll
for
try
that.
No
Thank
problem.
you.
You're welcome.
Okay. I'm going to sign off for now, and any more questions that we have, let's answer at lab at 12:30. This way we get a break and eat some lunch and come back, because I'm starting to feel a little brain dead from this morning. I ate breakfast too early. Okay. All right. So I'll see you at lab.
Thank you.
Thank you.